Good afternoon, everyone. I'm Jean. Today, on behalf of Helios, I'm presenting to you a new approach to treat cancer metastasis. I probably don't need to emphasize how cancer is a leading threat to human life. Indeed, in the last decade, we have significantly improved the 5-year survival rate to as high as 86%, as you can see in the, actually, I'm controlling that, sorry. As high as 86% as seen in the breast tumor indicated in the orange bar. However, at the same time, cancer treatment still relies heavily on early diagnosis. Whenever it metastasizes or diagnosed at later stage, the 5-year survival rate can drop significantly to as low as 4%, as indicated in the lung cancer. In 2016, Helios founders reported a clinical observation which shows a strong correlation between cancer relapse time and a structure called NETs. When the patient has low NETs level in their blood serum, which is indicated by the orange bar, the relapse don't happen until more than 5 years. However, in the high NETs patient groups, more than half of the patients develops relapse of tumor within 8 months. This sharp contrast actually suggests by controlling NETs or inhibiting NETs, we can potentially delay the relapse of tumor. In 2009, for the first time, Helios founder Dr. Yanmin Wang reported that NETs formation actually requires a protein called PAD4. And PAD4 inhibitor is able to abolish the formation of NETs. We then tested the inhibitor in mice. To evaluate the inhibition of tumor growth, we used a triple-negative breast cancer model. And in the control group, you can see we observed a formation of tumor nodes, and its red dark color indicates angiogenesis and malignancy. However, when PAD inhibitor is applied, tumor nodes shrink to almost nothing, and its pale color indicates less malignancy and less angiogenesis. To test the inhibitor for liver metastasis, we use a liver metastasis mouse model. And compared to the control groups on the left, where liver tissue is essentially buried in tumor nodes, PAD4 inhibitor is able to decrease the tumor mass by 73%. And that is comparable with what's been observed in the PAD4 knockout mice. So in Helios, we have collected essential pieces to success. We have a founding team with KOL in the field with a combination of expertise in oncology, biochemistry, and chemistry. At the same time, we have a strong advisory team with professionals from big pharmas with more than 20 years experience covering the expertise in oncology clinical trial, CMC, liver surgery, and cancer metastasis. Last but not least, we have a patented first-in-class technology with a potential value of up to $65 billion. And this is the story of Helios. Now I'm happy to answer any question you may have.
Jen, I'm sorry, go ahead. Jen, just to be clear, so if Helios owns the application of the PAD4 in cancer. Mm-hmm. So if nobody else could use it for metastatic cancer.
We actually have a composition of matter patents, which patents the exact structures. And that actually gives us a freedom of operation in the field. And indeed, we covered its application in cancer and autoimmune diseases. And that's where the $65 billion market comes from.
Okay.
Yeah.
These studies kind of reminded me of my PhD project. I was working on Nevavar and melanoma tumor models, so interesting results. So one question, if I may, based on this pathway that you're going after, what can you combine it with synergistically? Is there something that you've identified in the pathway that you can combine it synergistically to get more efficacy? So are you looking at any combination therapies at this point?
That's a great question. You know that if I have enough time, I can show you that PAD4 is actually a protein that's been highly expressed in immune cells. So we all know that immunotherapy is a hot area nowadays. So we are looking forward to combine PAD4 inhibitor with potential, PD-1 or PD-L1 antibodies or any IO therapies, along the way.
Gotcha. So you're starting to do some of those in my studies.
Currently, we are trying to, claim a drug candidates. So we haven't launched the combination therapies yet.
Gotcha.
But yeah. And at the same time, PAD4 is a biomarker for clinical trial.
Gotcha.
Yeah, so can you speak some more about what stage is the company at, and what do you expect to accomplish in the next year or so?
So currently, we're doing a planned lead compound optimization, and we are aim to claim a drug candidates by end of 2018. And in the next 1.5 or 2 years, we're gonna run GLP studies and to file an IND application.
Jin, on the padlock parallel that was acquired by Bristol Myers Squibb, do you know how much they raised before they exited?
Padlock is founded in 2013, December 2013, and they raised $33 million, and they spent $18 million in a year before they are acquired by BMS. And we believe that most of the money are spent to do high throughput screening of inhibitors. As far as we know, we didn't see any reports on GLP data for sure, but we don't know if there's non-GLP data on toxicology or ADME, PK. Yeah, probably there will be.
Do you think you'll need a similar amount of capital or somewhat less?
No, because we don't have the plan to do any high throughput screening. Our lead optimization is targeted, and we know exactly which group we want to modify. I mean, the founder team has working on PAD4 for more than 15 years now. So we know very well where potentially of the inhibitor can be modified, and probably some other part we cannot move. So it will be planned and very selective optimization of the lead compound.
Thank you.
Are there any possibilities for non-dilutive funding, grants?
We tried to apply to SBIR last year, and we didn't get one. And hopefully we can get this, prize as non-dilutional fund resource.
Excellent.
Yeah. And by the way, we actually get some, investment, equity investment into the company, so we can actually launch a little bit of, drug development.
Just one other question. Where else is PAD4 expressed? So when you're using an inhibitor, what kind of side effect are you forecasting?
So actually PAD4 only, I wouldn't say only, PAD4 highly expressed in immune cells such as neutrophil, macrophage, mast cell. That's what we have known. We are eager to know whether it expressed in T cell or B cell. We don't know that yet. But we know that in somatic cells, they don't express. Yeah, so naturally PAD4 kind of provide a selectivity itself because it doesn't express. And also we are running some tests to, we want to see whether PAD4 is also highly expressed in cancer cells. And there are papers published about clinical observations that link PAD4 high expression with malignancy of cancer.
Okay, thank you. With that, we're out of time.
Thank you.